Good evening, everyone, and welcome to third day. Right? Just a minute. Definitely, we start in a minute. All right. So let's start with our today's topic. That's the brief introduction. Okay. So for today, what I have planned is that we'll be starting with the basics of nervous system, right? And then gradually we'll be understanding those core concepts which will be touching physiology. So when we'll be learning physiology, at that point we'll be expanding on it, and then we'll carry it on all the way to the pharmacology, right? So that's how things will go. So it's like, I think, hope everyone is getting those notes properly, right? Those PDFs from that shared folder. In case of any difficulty, just send me a mail at that, the mail address, still you are mbbs at gmail.com, right? So without any delay, let's start with our today's topic. And this is the nervous system, which we'll talk about. Now, in this nervous system, so we'll try to simplify it as much as possible, and gradually we'll go all the way to those white rami communicants and gray rami communicants. How exactly, say, except receptors, will be touching almost every part, right? In the nervous system. Receptor is something which we'll be taking into the physiology, because there is lots and lots to learn about the receptors. So in nervous system, simply speaking, we divide it into two parts, say CNS and say PNS, right? Central nervous system. And central nervous system will be comprising of brain and the spinal cord, right? Quite simple. In case of PNS, we do have two parts. One which is voluntary, second which is not under our control. So one which is voluntary, we call it a somatic, and something which is autonomic, we call it ANS, or the autonomic nervous system, right? And then this is divided into, say, sympathetic, and I'll write in bit short, say, parasympathetic, right? But apart from that, yes, we'll be elaborating more on sympathetic, parasympathetic all the way. But apart from that, both of them, they would talk to each other in the form of enteric nervous system, right? It is in the form of enteric nervous system, NS, enteric nervous system, something which is in GIT, right? And this is like a very interesting thing, because if the clarity for this enteric nervous system is not clear, then it keeps on bugging almost during anatomy, during pharmacology. That is, what about all those superior mesenteric plexuses and inferior mesenteric plexuses, and that celiac plexus, from where they come, where they go, right? It keeps on annoying. So we'll clear all those concepts, right? Okay. So this is the thing. Next, whenever we deal with afferent and efferent, right? Afferent and efferent. E is for exit. Exit means it is going, right? It is going. Everything is with respect to brain. So anything which is going from the brain, right? It is going out. So that means we'll call it motor, because it will be going to some muscle, it will be going to some target and will act on it, right? And whatever the brain is receiving, right? So obviously that would be the sensory. That would be the sensory. So in response to afferent, there would be reaction on the path of efferent. Right? So far, so good. Always remember, whenever we talk about sympathetic system, sympathetic is always thoracolumbar, right? Thoracolumbar, right? In between. And to be very specific, it is T1 to L2. And then the parasympathetic is craniosacral, right? Two ends. So we call it craniosacral, right? Now in cranio, once again, you have to be a bit more specific because these are the cranial nerves over here. So then you add 3, 7, 9, 10. These are the cranial nerves which we'll be dealing with. Right? And when it comes to sacrum, it is S2, S3 and S4. That is like that sacral plexus, and thus it makes the entire system. So putting in simple words, thoracolumbar is the middle section and craniosacral, they are the extremes. As I said, we'll be talking about even ENS also, right? That is enteric nervous system. Now it is worth understanding, right? It is worth understanding. This enteric nervous system, right? I'll just write enteric so that it remains in the mind. This enteric nervous system, it is like intrinsic nervous system, right? Say it is like intrinsic. For nervous system, I'll just write NS of GIT. This is intrinsic nervous system of GIT. So that when the GIT is working, whatever the response, which say is this system is getting from the GIT organs, right? Immediately it won't go to brain all the way and will start doing the function. Everything will be done at that point only. So input and output, they both are present over there only, right? So in simple words, we just say that it is like a reflex local circuit, right? This is like a reflex local circuit, right? Circuit is CKT, so that is circuit, okay? And then this ENS, right, will talk to CNS that, okay, this was the problem and I managed and this is what I did, right? So they both keep on talking with each other. So that's why this ENS is also known as, aka is also known as second brain, right? It is also called as the second brain, right? Just a minute. Sorry. Okay. So it is also called as the second brain, right? So what happens that in the form of that input, right, this is like, say, physiological conditions of GIT. So that would be going into this system as an input, right? And then it gives the output, and that output is in the form of whether you want to control the gut movement, whether there is any fluid exchanges needed, whether there is any increase or decrease in the blood flow, etc. Right? So all the movements, all the blood flow, secretions, right? Those things, they are in response to that input. So this is like a local autonomous system. This is what is called as the enteric system, right? Local autonomous system or local autonomous function. Yes, good evening. Now in this ENS, if you dive a bit deep, right, you'll find that this ENS is actually very extensive, right? There are more than 20 different types of all those neurons, right? Enteric neurons. And all these enteric neurons, when we say that they are more than 20 different types, means 20 different functions, means by their functions, right? There are over 20. So many of them. And they can be divided into even 3 classes also in general, right? This class 1, then there is class 2, and then there is class 3. Say class 1 is sensory, class 3 is motor, right? So over here, this is sensory. So very easy to remember. This is sensory and this one is motor. But then there is something very interesting. This is class 2, which is called as the interneuron, right? This is interneuron. So in case sometimes if there is any need of those shortcuts, so then these interneurons, they come into picture. So over here, in case of sensory, you have got all the plexuses, right? Like say, when we'll be learning histology of GIT, you'll find that there is myenteric plexus. Then there are like say submucosal plexus, right? All those submucosal plexus, right? Such plexuses, right? Such groups. So they all will be working, and then that's how the response is received, right? Total number of neurons, they are in millions, right? Huge number. Okay. So coming back to this, when we talk about nervous system, let's learn the nervous system right from the basic, means all the way from say the structure or the formation to the spinal nerves, to the sympathetic parasympathetic, to the neurotransmitters, and then we'll stop only at that point when it goes into the field of physiology, right? When it goes into the physiology. Yeah, that's okay, but it is like not everyone was present. So don't worry about it. And in fact, so many new things will come. So when it comes to say nervous system, this is neuron, and then neuron is something which is conducting the transmission, right? But then there are few non-neural cells also, right? They are like non-neural cells. So those non-neural cells, they will be doing only one function, that is the support, plus something more, right? They are called as the glial cells. So we'll be talking about both of them. Now these neural cells, when we say that they are polarized, that means they would be sending the current in one direction, right? And this is like they transmit the signal, right? So there is a signal transmission. Most important that the heart and the brain, they are like once damaged, gone forever, right? Because they can't divide. So these are like called as the permanent cells, right? They are permanent cells. So this neuron, which is like a brick in the entire building, right? So this is the smallest functioning unit. So it has got the say the body, right, which is also called as the soma, and then there is axon, and then there are terminals, right? So that's how it is divided. Okay. So in case of neuron, right? In case of neuron, if we just draw it simply, right, it is something like this. So here is right a normal neuron. In this neuron, say there are few granular structures, right? These granular structures, they are called as the Nissl body. And the most important function of the Nissl body is protein synthesis, right? They take part in the protein synthesis. They are the site of protein synthesis, right? So this is one important point. That is the protein. And why they are granular? Why do they really look like it? Because it is the rough endoplasmic reticulum, right? Rough endoplasmic reticulum. And why it is rough? It is rough because of ribosomes. So these are the important sites where the protein synthesis will be going on, right? So over here, this is the body, right? That's the body. These are dendrites, right? These are all dendrites. Dendrites. And this one is called as the axon. Axon. And finally, we do have over here, these are the terminals, synaptic terminals, right? Or terminal ends. Now, interestingly, any part of the neuron can talk to any part of the other neuron. So ideally, right? Ideally, it is like, say, this is head, right? This is axon, and these are the terminals. So then there would be another dendrite over here, and then there'll be another axon, and like a train, right? It keeps on going. This is very ideal situation. Actually, things won't be going exactly like this, right? There can be so many variations. The direction of transmission would be in this direction, that is from head to toe, right? Focus on this small portion. These Nissl bodies, they will not be present after a certain point, right? And that point, this is what is called as the axon hillock. Axon hillock. Nissl bodies, no Nissl bodies. So I'll just write Nissl and I'll cross it. That means it is not present from this point onwards. So over here, this is that point, that is from where things would start finding the voltage would start passing. That is, the action potential would start. So that's why this action potential, how those sodium pumps they will work, everything is what will be picking up in first 2 days of the physiology, right? Along with all the receptors. So things would be crystal clear. Yes, you are right that this quite a small portion of it was already taken into one of the class of that neurophysiology, right? Neurology. But over here, as we are trying to compile everything, right? So I don't mind taking it again, because again, even if you attended that class, something new will definitely come even in today's session, right? So because as we are writing, so surely new things will come up. Okay, so this part is what is called as the trigger zone, right? Trigger zone. Now, as we say, that trigger zone is what? Trigger zone is from where the voltage would be starting. And how it starts in this case? Over here, in case of nerves, we need to remember that it is the voltage gated sodium channels, right? They open. Now, in case if you have got the doubt about that, what are these channels, right? What is this voltage gated and what exactly is sodium channel? Putting it into very simple terms, because you have to just wait for the day when we'll be dealing with actually these channels and the physiology part of it. But to make things easier, see, in our body there are so many ions: sodium, potassium, chloride, etc. Right? Now they just cannot walk in and walk out of the cell. So that's the cell membrane, right? Which is like that phospholipid layer, biphospholipid layer. But I'm not drawing it. I'm just keeping it simple that these ions, they are not permitted that they go in and out as per their wish. No, there is a control. So that means there are few pumps. Those few pumps, they will be pushing these ions out or in, right? Even against the gradient. Gradient means whenever this is the high concentration area and this is low concentration, things would be going from high concentration to low concentration, right? In which no energy is needed. It will just travel. These pumps, they will be going against the gradient and also they'll push it, right? So this entire game is of those pumps. So here, just think it that way, that this is a special pump, right? This is a special pump which is operating on a specific voltage. You must have seen those lights that as soon as the sun sets and those lights, they start glowing because they are light sensitive. So we'll call them that light gated lamps, right? So as soon as light falls on them, right, they shut down, and when there is less light, so they will automatically start. So such things, it's like electronics. So same way over here also, this is like nature's electronics, and these pumps, they are like at a specific voltage, right? At a specific voltage, they open up, and then sodium will be allowed to pass through it. And when sodium will go, then starts the entire story of those action potential, how those potential they generate, how the spikes came, and similarly, say what really happens in nerves, what really happens in heart, right? How are they both different? All those things they will be covering up in physiology part. But today we just understand that these are the channels which will be working accordingly, right? Okay. If we take a cross section, so that nerve fiber, right? This is a nerve fiber. So this nerve fiber, it has a coat, right? And that is what is called as the endo inside, right? So it is called as endoneurium. Then such several nerve fibers would be there, and this whole thing is called as the fascicle, right? So then there is a connective tissue which will be covering them. So this entire system inside is called as the fascicle, right? It is called as the fascicle, and this fascicle is covered by what's called as the perineurium. Right? This is also called as like blood nerve barrier. So that's the reason it plays quite a major role when you are going for any of those surgeries. So it is a connective tissue. I'll just write CT, connective tissue. And then there are several such fascicles. So several such fascicles, right? Several one. And then there is a thick connective tissue which is covering all of them, right? And this thick covering, this is thick connective tissue. This thin connective tissue is called as the epineurium, right? This is epineurium. Okay. So that's how it went from endoneurium to perineurium to epineurium. Yeah, that's right, in light sensitive. Yeah. So now coming to those glial cells. Those glial cells. You know, friends, today I saw one very interesting thing. Means it really doesn't matter, but say as I was about to start today's session, I just saw before starting the session there was one dislike. Means even session was not started, still there was one dislike. So I became a bit curious that who is this gentleman, right? At least Baba sun to lo, right? Anyway, doesn't matter. That's absolutely fine. Okay. So coming to these glial cells, they are also called as the neuroglia. Right? This is just another name. Important is like say in circuit, right? In our house there are all those electric wires, but those wires, they are not hanging in air, right? There'll be few boards, right? Either the plastic board or there'll be say wooden boards, and then few screws will be mounted, though the current will be passing only on those main wires. Same way over here, right? Those neurons, right, they are like active current, and then this infrastructure which we are about to discuss. अरे 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 no no I say Janvi, it was not, I became curious because before starting the session, right? That's why I became a bit curious. Okay, so neuroglial cells, they are, I would say in one word, they are non-neural cells. Non-neural cells, right? So in that case, their function is just to support, right? That's the best thing. One is the support. Now think of your house, right? Actually the wire, which is that copper wire, right, that is what is carrying the current, but then surrounding it, then there is, depending upon, let's say if it is a wire for AC, so then that wire would be thicker, and then there will be a thick plastic coat, right? And if it is a thin wire, so then there will be thin plastic coat, but it would be there. Why it is there? Because when one or two or three wires, even if they are lying on each other, nothing happens, no short circuit, because it gives insulation. Same thing is over here. So it gives protection, it gives insulation, right? It gives insulation, and always remember these words. They maintain homeostasis, right? Homeostasis. In human body, homeostasis is so important, because in our house, so we can say, okay, chalo, this wire has eroded, let's change it, right? We can't go and tell to God, okay, now this radial nerve, I think it is a time to change, give me a new radial nerve, right? It won't happen. So that's why it is very much important that the system should be so well defined that it should keep on running for years and years and decades effectively. And that's the reason that all these various types of glial cells are there. One cell, right, which we must know, and it is astrocyte. So again, this astrocyte is like, it is so much abundant that in case if anyone asks about the glial cells, blindly if you say that, okay, answer is astrocyte, mostly you will be right, because it has got so many functions, right? It is most abundant, right? In other words, what I'm trying to do is, I'm giving you a hint that I may ask you a question about astrocyte, right? Because it is, yeah, they provide nutrition also, exactly. Yeah, right? So they are most abundant, right? And they are, as you said, yes, nutrition, right? So let me write it. They provide the nutrition also, and they provide, say, support, right? So in other words, we can say that they provide physical support as well as the metabolic support, right? So that covers everything. Plus it is acting like a storage, right? So that is in the form of potassium buffer, right? It keeps the potassium buffer. And this point is interesting. Though they are not part of that electrical system, but any excess neurotransmitters, right? Any excess neurotransmitters, they are removed by them. So I like this point, very interesting, right? So that's why I'm marking it. So they do this. So if astrocytes, they are most abundant, right? Large number of projections, and they are like so active, so many of them. So just the reverse is those microglia. Name itself is, see, microglia, right? Micro, right? They are less in number, right? They are small, less and small. But these microglia, they feel that okay, we are small, but we have got big function, right? They are like our Pac-Mans, right? They are phagocytic cells, phagocytic. So whenever it comes to like those scavengers to clean up the things, they will enter into the system, right? Third and fourth cell, I'm writing it like this. Reason? One, what is called as the Schwann cell, and second is oligodendrocytes. Oligodendrocytes. So in Schwann cell and oligodendrocyte, they both will be going towards one function, and that is myelin, right? And as we'll be discussing myelin in depth, so right now I'm putting those Schwann and oligodendrocyte just as it is, right? Because we'll be discussing at length. Simply putting, Schwann cell, what they are doing, they are making those myelin sheaths, right? In case of peripheral nervous system, and these oligodendrocytes, they will be doing the same function, but in case of central nervous system. That's the only difference. Though there are few other differences, but we'll see as we'll be talking about it, right? And apart from that, there'll be few others, but that's okay, right? Like say those tennisites, so which is for transport. Then that, let's say that that choroid epithelium, right? So that choroid epithelium, right? Its classical function, CSF secretions. At this point of time, we are not giving much importance to this choroid, etc., because CSF itself would be a very, very important thing. Why to remove neurotransmitter? Well, that's what I said over here. Excess, normally not removed, right? But in case if it is excess, so it is like a defensive mechanism. So that's the excess part which is removed. And this question which you have asked, right, why to remove the excess neurotransmitter, it is one of the most interesting question, that these neurotransmitters ज़्यादा भी खराब कम भी खराब, right? If they are less, so that required action won't occur. If they are more, then that action will keep on occurring on and on and on, and that will lead to all types of disturbances, right? Excess of both the ends is bad. So that's the reason that these neurotransmitters, they are so important, and we'll be talking more about it. Regarding the blood-brain barrier, yeah, blood-brain barrier is, say, its importance will come when we'll be dealing with, say, in pharmacology. Say here is the blood and here is the brain, right? They both are separated by this membrane, right? And if the drugs, those drugs which can cross the blood-brain barrier, that means those drugs, irrespective of where they are working, right, their primary target, but they will be having CNS side effects. CNS side effects, right? Because they are entering into the brain, and once they enter, they will be affecting. So that's the reason blood-brain barrier, that is very important. Similarly, when it will come to the placental barrier, that is also very important, right? So, and in pharmac, these things will come in the form of when we'll be talking about the side effects, right? Regarding the neuron, as we saw one very ideal thing, right? But these neurons, they can be actually of three types. One, what is called as the uniaxial. Now this uniaxial, normally we don't see much, right, in human body. It is classically seen in large amount in case of what's called as the invertebrates. Now what are these invertebrates? Invertebrates means those who are having no spine, right, no backbone. So in those cases, they don't have any backbone, right? This is one. And such creatures, they are cold-blooded, right? Nothing to do with murder, right? Right? It is not like that they are murderers, but cold-blooded means the temperature of their blood, right, it keeps on changing with the surrounding environment. So they can live on the land, they can go underneath, right, temperature is changing, so that's not a humans. They are like warm-blooded. So even if you are on the Everest, if you really check your blood temperature, right, it would be almost the same, right? CSF secretion, cerebrospinal fluid, is very important for one simple reason: nutrition, right? Then, though there are so many other functions, but the nutrition part. And there is a complete system of that CSF secretion in the form of lateral ventricles to third ventricle to fourth ventricle, and then from there it will go all the way into the spine, right? So that it can properly provide nutrition all the way to our central nervous system, right? That is the brain and the spinal cord. See, that's the reason that whenever there is any of the infection, right, it is the CSF tapping which is done, and then it goes for the lab test, right? Yeah. So in these invertebrates, all those crabs, spider, worms, insects, right? All of those, they come into invertebrates, right? So like say those crabs, spiders, right? Insects, right? Makhi, machhar, all those things, they come into these invertebrates. And in such invertebrates, right, their neuron, right, definitely they'll be having neuron. So their neuron is like, that's the cell body, right? And then there is only one projection, right? There are no dendrites, right? And only one terminal which is coming out, and it is called as the neurite. It is called as the neurite. That's it. This is the cell body. Now in humans, it is there, right? In humans, right? But only in very less quantity. This is, they are always called as the sensory ganglion, right? Those sensory ganglion in humans near the skin, right? Near skin, they are uniaxial, right? Then there are bipolar, right? They are bipolar. These are like uniaxial or unipolar, right? We can also say unipolar. These are the bipolar. So in bipolar, this is cell body, right? And then there is axon, and then there is one dendrite. That's it. So here is the axon, here is one dendrite. Dendrite means something which is coming out from the body, right? I read somewhere that in fetus uniaxial is present. Possible, but honestly, I don't know. Right? I don't know. I'll let you know tomorrow. Right? I'll figure it out if it is really there or not. But at this point of time, I don't know. But I'll definitely tell you. Okay. All right. Right? I'll let you know tomorrow. Okay. Okay. In fetus there is unipolar that is given in NCT. Okay. All right. So you must be right. Acha. So coming back to this bipolar, this is not in common, not very common. Again, this is also not very much in common. Bipolar we get it at two places. Yes, what you said is right. One is say olfactory, that is for the smell, and in some portion of retina, right? Some part. Otherwise, this is 1 and this is 2, this is 3. Apart from all these things, they all are multipolar, right? That means all the dendrites, right? All those. So all except 1, 2, 3, they are multipolar. So this is okay. So this is how, say, they are really formed, right? Now we pick up that point, that is the myelination or the myelin. This myelin is interesting. First, that this is white in color, because it has got lipid. So this lipid is giving white color. Now, when we draw it like this, that this is the axon, right? And that's how this insulation process is going to occur, right? And these cells, they are called as the Schwann cells. Schwann cells. So in PNS, this is Schwann cell, and same thing if you are dealing with say CNS, so they are called as the oligodendrocytes. Oligodendrocytes. Okay. So we'll be talking about in detail about Schwann cells, but purpose, say in CNS, it is almost the same thing would happen, right? So purpose is the insulation plus it increases conduction speed, right? It increases conduction speed. Actually, it is like that those impulses, they jump like this, right? They travel that fast, right? It improves conduction speed. Now, when we talk about their development, so way back, right, in the third trimester, it starts its formation, right? This is like formation. Its formation starts in the third trimester and it continues for almost 40 years of adult life, right? So long. Regarding motor and the sensory, so this also indicates that all those formations, right, they keep on changing. That is that support cells, they are getting generated. For the motor, it is over in the first month of life, and sensory also, they are completed by 6 months. So within this time frame, at least all those nerves which are to be needed for the myelination, they are done. For peripheral nervous system, right, they give strength, they give, as we said, metabolic support. It gives metabolic strength, and metabolic support is what is given by these myelin. So that's in brain myelin. Yes, I'm coming to that, right? I'm coming to that regarding that where exactly this myelin is present, where exactly it is not present. Just hang on for a minute. Okay. So in case of ANS, autonomic nervous system, right, you will find that they are non-myelinated. So axons in ANS and some small sensory fibers which are for pain, right, these are non-myelinated. So when they are non-myelinated, so I'm writing it like this, right? They are non-myelinated. The process is quite interesting, that here is the axon and here is, let's say, that Schwann cell. So this axon, and right, it invaginates into the Schwann cell. So then the situation becomes something like this, right? Like this. And then this whole cell, this Schwann cell, it starts rotating. So when it rotates, so obviously here, so the situation would be like, it becomes like this. Then it still rotates more, it becomes like this, and it keeps on rotating. So finally goes round and round and round and round, and that's how the multiple layers, they are formed, right? Multiple layers are formed. And then these spirals, right, these are called as the spirals. And these spirals, then there'll be layers. If you see, there'll be one layer, then there'll be second layer, then third layer, right? That way layers will be there. And in between these layers, there is now lipid which is getting deposited. So lipids, now they are in between these layers. Very important to remember is sphingomyelin, right? Sphingomyelin. And that's where you'll be getting all those lines like minor dense and the major dense lines. So, right, the situation would then finally be something like this, that yes, there would be all those concentric circles, right? In between, you'll find that there is all those, the lipid and the protein layers, they are formed, right? So they all get fused. And this is one Schwann cell. So when the axon is like this, right? So that Schwann cell, one Schwann cell, which rotated, and then it formed this myelin sheath. So this myelin sheath would be covered all the way, right? Then there would be the second cell who will be working like this, and it will also be covering the Schwann cell from surrounding it, right? And then the process continues. So as we said that they act as insulator, and at the same time they prevent any of the leakage of ions from any of the direction. It means it won't allow anyone from the outside to come in, and it won't allow the inside ions to go out. Right? This leads to what's called as the pure signal. Right? It will lead to pure signal. Otherwise, there would be lots and lots of all those interferences. Exactly like that nowadays, so because of that ELCB and MCBs, things are much better. Else, if someone in the building is using that mixer juicer, and immediately you will start finding that there are few disturbances in the television, right? It used to happen. Nowadays it doesn't happen, simply because now everything is isolated because of those ELCB, earth leakage circuit breakers and miniature circuit breakers, right? Okay, so here that's what is called as the node of Ranvier, right? This node of Ranvier. And if I draw it like this, so like this, like this, and let's complete it. So then the all the way from here to here to here, okay, this is what is called as the internode, right? It is called as the internode. To give you a brief idea, the size of this internode is about 150 to 1500 microns, right? 150 to 1500 micron. So how much this micron is, right? Micron is, say, 10 raised to minus 6 meter, right? Meter. 10 raised to 6 means it's like it's 1 millionth part of meter, right? And you know our hair, right? Our hair, human hair, it is 70 micron thick. So that's just to give you the idea that how much this internode will be, right? So they give those pure signals. These myelin sheaths, right, what we talked about is very ideal, right? Very ideal. But sometimes those adjacent layers, they don't fuse, right? Those adjacent layers, right? Adjacent layers, they don't fuse properly, right? Maybe because of some deficiency, etc. So in such cases, if this is the axon, right, you'll find some oblique clefts, some oblique lines, and that's what is called as the Schmidt clefts. Schmidt clefts, and then that's the extra you have to remember, Lanterman. Right? So Schmidt-Lanterman clefts. And in case if there is any injury to the nerve and nerve is getting degenerated, so obviously size of all these, they will start increasing, right? They will become bigger and bigger because, and when the nerve is getting degenerated due to injury, it is called as the Wasarian degeneration, right? That's, and there is lots to learn about this Wasarian degeneration. Right, so many stages, etc. What really happens? Roughly, you can say that it starts within say 12 to 36 hours after the injury. If the nerve is injured, this degeneration starts within a day or 1.5 day, and it takes about 12 to 15 days before the entire thing is degenerated. So 12 to 15 days long procedure it is, right? So that is what is called as the Wasarian degeneration. As we said that oligodendrocytes, right, oligodendrocytes, they are also behaving quite similar to these astrocytes, right? Quite similar. But just one important thing to know, that over here it was one cell which was coming close to that axon, winding around it and was making all those wraps, right? So it was wrapping around. In oligodendrocytes, they are bigger ones, right? So what they do is one cell can actually wrap many axons. That is what happens, right? So that's how it can. So that's why this is about brain. So when one oligodendrocyte is holding so many axons, so obviously all of them, they would be remaining together. So that's what really happens in case of this. Now, this takes us to next step, because from there we can talk about the neurotransmitter, right? This is what's called as the snap, right? That is what is called as the snap. Now in this snap or the synapse, right, it is like where two neurons, they are talking, right? That's the point where these two neurons, they are talking with each other. Now we said this, right, that this is one neuron, and this neuron, it can really, any part can talk to any part. Yeah, actually it is like that. Though some things are rare, right, but actually they can. So see how interesting names are given, right? Nothing bigger, but as I say sometimes, that in case if there is any fight, right, you start speaking these names, right, or your opponent will automatically run away. Like, say, we just give the name. This is soma, right? So this is also soma, correct? So now, when these soma to soma communication, it occurs. So somatosomatic, right? So it would be called as the somatosomatic synapse, right? That's where they'll be, but it is rare. It is rare. Or when this dendrite, right? This dendrite, right? He goes and communicates to axon, right? So dendritic axon communication, again a rare thing. So that way, when the dendrite is communicating to say axon, or when this soma is communicating to this soma, that is rare. Or when dendrite is communicating directly to the soma, right, that is also rare. So these are some of the rare ones. But otherwise, classically, it's all like that train, right? Somatic is what it is communicating with, say, those dendrites to body, to axon, to terminals, and then these terminals, they are having that, say, synaptic formation, and then from here things would continue to the next neuron. This portion is the synapse, right? If we really zoom it, right, so then it would be like this. It would be like this. So here, this is synapse. Anything before synapse is called as the presynaptic, and after synapse is the postsynaptic. Right? Simple. And in between, it is the neurotransmitter which will be working. It is the neurotransmitter which will be working in this space. These neurotransmitters, they can see, neurotransmitters, they don't have any bad intention. They said that we are made for a certain function, and that's what they do. So that's the reason that those postsynaptic potentials, they are increased or decreased because of all those neurotransmitters. What would happen? That these neurotransmitters, some of them, they are designed in such a way that what they would do, they would increase sodium influx, right? When the sodium is increased, so that is excitatory, right? And things would fire up, so that is excitatory. There are few who will increase the chloride influx. Now this is like a complete inhibitory system, so that's why this is like a reverse switch. One is to stimulate, second is to inhibit, right? So for example, in neuromuscular junction, when such things happen, those muscles would contract. So this is like neuromuscular junction. So for that, it makes complete sense. Yes, it is good because it is needed, right? What about this inhibitory? Say for example, GABA, right? Whenever it would come like GABA, which is gamma amino butyric acid, right? It is butyric acid, gamma amino butyric acid. So whenever the gamma would come, its function is slow down or the stop. So this means we'll be calling, for us, everyone is important, right? This is like accelerator. So in that case, this is like those brakes, right? They both are needed to drive successfully. So in case if our car is going at a very high pace, so yes, we release the accelerator, we press the brakes. Same thing would happen. And if you just press the brakes and even if the accelerator has been pushed, yes, car would topple, right? If you don't apply the brakes, so then car would keep on working. In fact, drugs will work exactly like that. So sometimes you have to apply the brakes and you have to take away the accelerator, right? So that's how the neurotransmitters will be handled. Yeah, when the nerve is injured, that is Wazirian degeneration. That's right. Right. So over here, for the synapse, which is that junction when there is communication between these two nerves, right? So there will be of two types. One, where there are chemicals which are used. Second, in which there is electrical system, right? I used to say electronics, right? But no, there is no electrical, because we'll be dealing with ions. So in case of chemical, they are bit slow, right? They are bit slow because that chemical will go, right? And how those neurotransmitters, they actually work, right? It means in this pre-synaptic, there are all those vesicles. Those vesicles, they will travel near the surface and then they open up and then there will be release of these neurotransmitters. These neurotransmitters, they will go on to the postsynaptic surface and they will be acting on those specific, say, all those channels, right? Those channels would open, and then what this neurotransmitter is meant for, if it is meant for, say, opening up of sodium channel, so yes, sodium would enter and things would be like stimulated, and that thing would continue. In case if it is inhibitory, so then it will be opening up those negative ions, and that's how it will slow down the rest of the processes. But then these are like chemicals, right? But then there is something like electrical. They are found in heart and smooth muscles. So heart said, I can't wait for this much because I need rapid response. So that's where that there are gap junctions. Through these gap junctions, those ions, they travel, they finish up the work, and it is much, much faster as compared to chemical. Right? Chemical is also one way. Right? This is one way. So the way in this case, this current will go in only this direction. It cannot go in other way. While in case of this electrical, that is two way. Right? Current is two way. Now, in order to remember less and at times to get a very good clarity, right? I explained this thing even in my past lecture also. It's not here, but way back when we were discussing that neurology. That we said that those axons, right? Those axons, when they are myelinated, right? They are white, right? They are white. And those who are non-myelinated, they will be gray, right? But axons, they are myelinated. Those cell bodies, right? Those head ends, they are not myelinated, okay? So it means that those cell bodies, they are not myelinated, true? And that's the reason they would appear gray. So over here, watch this. A, straight line. M, straight line. W, if I write, right, it can be written with straight lines. So this means we now understand that axon, myelinated, are white, and these are what? These are cell bodies. See, this is curved. This is gray. You have to really write it like this, gray, right? In brain, now let's say in brain, what happens? Outer cortex, inner medulla, right? Outer cortex, inner medulla, right? So outer cortex, true, outer cortex, so easy. See, everything is in the form of all those curvilinear. While in case of, say, here, outer cortex, inner medulla, and this is the inner, right? So again, all those straight lines, right? So this gives us now clear understanding that in case of brain, the outer cortex is where there are cell bodies, right? Outer cortex is where there are cell bodies, and that's why it appears gray. And inner core is of axons, and they are myelinated, that's why they appear white, right? In spinal cord, things would be reversed, and now we'll be dealing with the spinal nerves in more detail, right? In case of spinal cord, right, I'm drawing it like this. So this is white and the inner one is gray. Inner one is gray. What does that mean? That means that this white, right, they are axons, and this gray, they are the cell bodies. right? They are the cell bodies. And it is this cell body which is actually the CPU, which is the processing unit, because axon is just conducting it, right? So here, in case of spinal cord, it is reverse than brain. Right? It is reverse than brain. That's what really happens. So this would be the gray, this one would be the white, and now you know white means axon, gray means cell body is CPU, that is where the processing really occurs. Right? Okay. This takes us to the actual spinal nerve formation. Right? So when we take a cross section, so we come across something like this, right? And then there is like anterior horn, there is posterior horn, right? This is like anterior, that's the posterior, right? And then we draw it. Right, okay. So now we are making the somatic, that is sensory. So this sensory, let's change this color. This sensory will come from the posterior horn, right? From the posterior. So that's where the sensory neurons which we are talking about. So that sensory would come. But see, whatever said and done, but actually they will be all going towards one common path, right? So over here, let's say it would be like this, and we just draw it like this, right? Let's change the color of this. Let's take it red. So from this spinal nerve, right, here comes this this is sensory, somatic sensory, it enters, right? Here it is. And then there are interneuron, and then there would be the motor neuron. So it will emerge from here and it will go and it will travel, right? That's how it would go, no problem. So here we'll call this as sensory and this as motor, right? This is motor and that one is sensory. This much is pretty easy. But then over here we have got what's called as the lateral horn, right? Lateral horn. That lateral one would be carrying this sympathetic supplies, right? Sympathetic, parasympathetic. So over here, when it comes out, right, I'm drawing it like this. It comes out in the form of, there is a relay center, right? So when it comes out over here, this is white rami. It is white rami. White rami because it is myelinated, right? Because it is myelinated. And from here, the postganglionic, once again, it will go into the spinal nerve. And this one is postganglionic and this is gray rami. So the point over here is that this is gray and this one is white and they are all dealing with autonomic nervous system, right? So simply putting, this is like a ventral horn, this is like a dorsal horn, right? Dorsal horn, ventral horn, right? And that's the spinal nerve. So this spinal nerve is carrying spinal nerve is carrying afferent, efferent, and even the ANS fibers, right? So that's how the nerve is really formed, and that's the reason that every portion is so important. Okay, moving on to, that's the brain, right? That's the spinal cord. This is cranial. This is cranial. Then we have got, say, cervical, right? Not in proportion. So then this is thoracic, lumbar, right? And finally the sacral and the coccygeal. Sacral and the coccygeal. Craniosacral, right? Craniosacral, they are for parasympathetic. So from the cranial, see, that's what really happens. There is always a formation of ganglia, right? There is a ganglia. But this ganglion, right, over here, the preganglionic fibers, they are long. So long preganglionic. And then there are short ganglionic fiber. And before, then they reach to the organs. But that's not the case when you'll be watching say thoracolumbar, right? In that case, but that's it. The preganglionic fibers are shorter and the postganglionic, they will be longer, and then they reach to the organs. This is what really happens, right? Long preganglionic fibers, this is because it has got the wider effect, right? It will be having a wider effect. Why wider effect? Because it is a long fiber, right? As it is going, it will keep on getting connected with so many, so it has got the wider effect. So parasympathetic, they are, right? Parasympathetic are craniosacral, right? Parasympathetic are craniosacral. So that's why parasympathetic is needed when you are at rest. When you are at rest, that is where the parasympathetic is needed. In case of sympathetic, right, there is ulta. Over here, there is short preganglionic. So when there is short preganglionic, it means it is for immediate effects, a very sharp focused effect. So this is for focused effects. Right? So that's how the difference between both of them. Right? So over here also, from lumbar region also, it will be like this, and then it will be going for those organs, and then over here craniosacral, right? They are with long preganglionic, and then the organs. So this is one part. Yeah, day-to-day life. So when we say the effect, right, always take it like that this is parasympathetic, right? It is for when you want to decrease the rate, you want to decrease the force, right? The way heart is working, right? Because it is like rest and digest. When it comes to sympathetic, right, it is increased rate, heart rate, increased force, right? That's what you get. Similarly, you are at rest, so these lungs, they are not requiring more oxygen, right? So no need to work. So there is bronchoconstriction. You need to fight in case of sympathetic. So this is like bronchodilation. dilation, right? Similarly, when it comes to eye, right, here there will be constriction of pupil, constriction. Over here in case of sympathetic, eyes are wide open, dilated, because we need to find out a path from where to run away, right? So that's how, say, these sympathetic and parasympathetic, they'll keep on working. No doubt, it acts on so many areas of the body, but yes, all of them, they will be dealing with all types of alpha, beta, alpha 1, alpha 2, beta receptors, muscarinic, nicotinic, all those receptors, right? At that point, we'll be talking more in detail. Acha. Few terms which I always say one should know. Whenever it comes to like mimetic, right? It is like, say, parasympathomimetic. That means it will be supporting or it will be stimulating parasympathetic, right? This is also called as cholinergic. Right? This is also called as the cholinergic because that's the neurotransmitter which is in use. And when you use the word lytic, so parasympatholytic, lytic means it will be inhibiting them. So it is inhibitory to, say, parasympathetic, right? Similarly, when we say sympathomimetic, so now you know it, right? Sympathomimetic, that means it is the friend of sympathetic, right? So it stimulates sympathetic. And similarly, lytic, that is sympatholytic, so that would be inhibitory to sympathetic system. So that's how things will be working in that group, right? And that's where we say cholinergic, anticholinergic. So if someone is supporting this, so it is cholinergic. If it is inhibiting, so then it is called as anticholinergic. Because finally, so it is the acetylcholine which is into use. Say someone who is supporting sympathetic. So in sympathetic, we say like adrenaline, right? There is so much of rush of adrenaline. So that is where they are called as adrenergic, right? And the reverse of it is anti-adrenergic. So that's how, say, we talked about today some basics of this nervous system. Now tomorrow my plan is to go for the embryology, right? Tomorrow we'll be dealing with some concepts of embryology, and then on Saturday we'll be conducting a test. So this time test would be a bit mild, and because it would be like from 4 days. But from next week, right? Next week we'll be having 5 days to study, 1 day to go for that test. Right? So that's how we'll be going for it. Okay. So today we are stopping bit early, right? Tomorrow's session would be definitely a bit longer, right? As say embryology is not only, say, complex, but there are so many concepts to really understand. So we'll be paying good attention to it, right? So we'll be starting with the second from 29th, I mean second week you're telling? That is from Monday. I don't know the exact date. Test topics is whatever we studied from Tuesday, and we'll study till Friday, right? we'll be conducting tests from that. All right. Yeah, it would be on Saturday. It would be on Saturday. Same, and the links have already been generated also, right? There are links. Today's topic is quite easy, right? So it was not much of the story to tell, but yes, for tomorrow definitely there'll be stories. Okay, right? So on Saturday it would be, the test would be at 7:30 p.m. right, IST. Okay, so 29th is Monday, na? Fine. So that's how we'll go for it. All right, this file I have already saved and I'll be placing it in our common folder, right? Thank you so much, and we meet tomorrow at 7:30. Thank you and bye-bye. Thank you.